All right, welcome back, AP Bio. Today we're talking protein synthesis or translation. In your textbook, if you're working off the 9th edition online, this is chapter 17. And I actually have an activity for you guys to work on and practice some of this. That will be kind of a discussion post on Schoology where we can send coded messages to each other. So I'll be explaining that at the end once we get through this. So if you haven't already listened to and completed the transcription lecture, now would be the time to go back and make sure you've done this one. It's on my YouTube channel, and it's also posted onto Schoology in the online resources, talking you through how we get from DNA to RNA and discussing the 5' to 3'. As well, the 5' to 3' was also gone over in the DNA replication. So definitely make sure you've got that because it applies again to what I'm doing down here. So let's work on first an overview of the 3 kinds of RNA that are involved here. There isn't just one kind of RNA. RNA being the nucleotides A, U, G, and C, but in the different forms it takes, it actually has different jobs. So one here that you're probably most familiar with is mRNA, or a messenger RNA. When you see BPs, I'm talking about base pairs. So mRNA coded from the gene is thousands of base pairs long, and it's got a start codon in there. Somewhere along the way, it's also got a stop codon. But we created that from transcription with RNA polymerase. rRNA, so a little r RNA, is ribosomal RNA. This is our ribosome, which I've colored in yellow. Over here, he's kind of got a little face each time. Ribosomal RNA, these are our ribosomes. It's our organelle. Remember that all organisms on the entire planet have ribosomes. Prokaryotes, eukaryotes, everything that needs to make its own protein. So that's our evolutionary connection. They're made in the nucleolus, which is inside the nucleus. There are 2 subunits. There's a small subunit, and then there's a larger subunit that attaches as well. It's not only made of RNA, but also proteins, and it's the most abundant form of RNA we've got. If you look inside of any cell diagram, you'll see it peppered with sprinkles, which are those ribosomes. Our tRNA, or transfer RNA, I like to think of this as the taxis. These are carrying the amino acids. So this is actually where RNA doesn't become exactly just single-stranded, but it sort of loops around and backs on itself, and it has hydrogen bonding that's forming some double-strandedness in there. At one end, it's got a specific amino acid, 1 of 20, and at the other end, opposite of that is the anticodon. So the anticodon, we would read them 3' to 5' so that it complementary matches the RNA going 5' to 3' as we read that. So we'll discuss that when we get into here. But this is to ensure that anticodon is bonding to the right codon on the mRNA to make sure we're bringing in the right amino acid. And how do we know that we connected the right ones in the first place? Well, there are these enzymes called amino-acyl tRNA synthases. Crazy word. Probably won't see this on the AP Bio exam, but these are specific enzymes that help to kind of bring together the right amino acid with the right tRNA anticodon. So they've got, an active site that is looking for the right structure of both of those, and so that we've assembled them in the correct way. So you can imagine there are 20 different or so tRNA enzymes that are building these. There are actually a whole bunch of amino acid codons. You'll see this in our codon chart that I will go to on the computer. So one of these that I'm posting on your assignment. There's a lot of redundancy, like UUU, UUC are both phenylalanine. It's that third letter of the code that has some wiggle room. So there might be the same tRNA that can carry both this codon and that codon to match, but they'll both have phenylalanine. So there's a little bit of flexibility with that third position. We call that the wobble. effect because there doesn't need to be as much specificity in the third position of a codon. So we have some flexibility in the code, which is great because you can make mistakes and have mutations that won't actually affect it. So notice here how the tRNA and ribosomal RNA, they've got pretty different structures. We've got a blob of a ribosome, an organelle, and we've got this tiny little looped strand of tRNA with an amino acid. These are very mobile. This is going to be a location where things are happening. And the code of the mRNA is a single strand, kind of like ticker tape, that's going to get read to decode the DNA message and get it into amino acids. So they are all made out of the same subunits, but they're structured very differently for their jobs. I'm going to talk real quick about these 3 sites on the ribosome and what their positions are. I don't think you're going to be seeing this on the AP exam, but it's just a quick intro before we get into the details. There are 3 locations within a ribosome, the E, the P, and the A. The P site is where the the tRNA that is holding on to the amino acid chain, like this, that's where it's waiting for the next one to come in. And the A site is where the incoming new amino acid is coming in, bringing in the next one. And then the exit site is where the one that just released its amino acid is going to leave from. So you can see like that. I kind of think of a ribosome as a love seat. It can sit 2 comfortably, but it can't really sit 3. So you're not going to have 3 tRNA shoved in there. We're always kind of like bringing one in as one is leaving out as well. And in your textbook, in chapter 17, you can see a little actual microscope image of this. That there may be one mRNA strand in the cytoplasm, but there are multiple ribosomes working and building the protein. Here's one that's early in the mRNA, so it's only got a couple amino acids coming out. Here's one that's farther along, it's got more. Here's one towards the end, it's almost done. So this is a way that ribosomes can maximize their efficiency and just follow each other down the mRNA. There's no reason, kind of like at Wild Waves, they don't wait for the person to get to the bottom of the slide before they let you go. They're just going to let people go the moment there's room. So I'll talk about amino acid structure and protein folding at the end, so we'll leave that down there. So if we are going to be building our protein, again, there's a few minor changes I've made on here that are different than the one that is scanned and put up on Schoology, but you can just follow along here and adjust as needed. I've kind of removed some of the things that I think were superfluous and not necessary from last year. So for the first step, we have already transcribed our mRNA. Let's assume it's in the cytoplasm, okay? And the first tRNA and the small ribosomal subunit are going to bind to the mRNA near the 5' guanine cap. And so it's looking to line up with that start codon. The start codon is not going to be at the very beginning. Remember, we've got some guanines over here at the end. I haven't drawn them in, but it's to prevent that degradation and chewing up the end of the mRNA. So the anticodon on the tRNA is complementary to the AUG start codon. So A is at the 5' end, UG is going towards 3'. So this is the 3' end of our anticodon and the 5' end of our anticodon. So UAC reading backwards to be anti-parallel, just like it would be if it was a double-stranded DNA helix. They kind of work in opposite directions. So these 2 pieces are going to bind. It's going to take some energy and initiation factors to kind of help get everything else together. So just how we had transcription factors helping RNA polymerase to bind and express a gene and get it going, we also have initiation factors here. They're not pictured, but just that's what kind of these like action lines are to kind of get everything going. So the large subunit is also going to bind, and the start codon AUG on the mRNA is showing us where to begin translation. That's not part of the promoter. The promoter would have been upstream from here on the mRNA. It codes for methionine, and this may not be the only AUG on that mRNA. 20 amino acids later, you might have another methionine. It always will start a protein, but it can also show up later in other parts of the protein as well. It's not just at the beginning. So our next chunk here, oh, by the way, this is initiation, and now we're talking about elongation. It's kind of the same 3 steps that I forgot to mention in transcription. Initiation, elongation, and termination. 3 stages of building either your mRNA or here building your protein. So our next part here is the ribosome's going to move along from the 5' end of the mRNA down towards the 3' end, and it's forming peptide bonds here between each of the amino acids. If you forget what the peptide bond is, I've got that down here in the corner. So remember, each amino acid goes NCC, NCC, right? An amine group, the central carbon with the unique amino acid R group, and the carboxyl group. And that carboxyl group bonds with a peptide bond to the next amino acid group. And you would be losing water in the process, because that's a dehydration synthesis reaction. This is all back in chapter 5, early in our biochemistry unit. You've probably got this in your notes. Now is a good time to review the basic structure of amino acids. I won't go into too much the different properties of these R groups, but I am going to review protein folding for you today. Okay, so energy is required to not only recognize and bring in the right anticodon on the tRNA to match the codon, but also to shift everything over and move. So you may recall in the Oculus, like the virtual like cell video that we did, where the motor protein is like moving, each of those takes an ATP. This is also taking ATP to move the ribosome and shift it over position to position and forming bonds. That's going to take energy too. So let's see, only the tRNAs that are complementary with anticodons will be accepted and be able to come into this A site. So I've been filling in the right anticodons that should be binding in here. You can do that as well. For termination and ending the process, we're going to be getting towards the end of our mRNA, towards the 3' end, and we're going to encounter a stop codon. There's multiple stop codons. There's UGA, UAA, and UAG. No, you don't have to memorize those, but you probably should have the start codon memorized, because that is the only start codon. Think of it like a race. They often only have one starting line, but you could finish a race at the 100 meters, at the 200 meters, there's multiple ways to stop. Stop codon is going to signal the end of translation. Remember that this is different than transcription, which had a transcription terminator. This is a stop codon. So start and stop codons for transcription, promoter, and transcription terminator for transcription. This last component, this is a release factor. It's kind of like a tRNA, but it's a little different. It doesn't actually have another amino acid attached to it. It's got a water molecule on the end. So rather than bringing in and binding another amino acid and forming another peptide bond, it's basically going to provide the water that's going to sever the bond and be able to close out the carboxyl group on the end here. So it's going to have an H2O on the C terminus, and the protein's going to release. With energy as well, action line, so more ATP needed. The protein releases, it finishes up its folding, and we've got bonding that's going to help connect things, and everything's going to come apart. So recall that you can find proteins not proteins. You can find ribosomes in 2 kinds of places in a eukaryotic cell. You can find them in the cytoplasm, or you can find them on that rough ER. So think about, if your protein is one that's going to work in the cytoplasm, you can form them on those free-floating ribosomes in the cytoplasm. The moment they're done, they're released and they're doing their job. But if they're proteins in eukaryotic cells that need to be transported to other parts of the body, modified and changed, like secreted in packages out into the blood, then those are going to be processed and formed on the rough ER's ribosomes. And I want you to go into chapter 17 of the 9th edition and look at page 343. There's a really great diagram there on how they get recognized for figuring out what kind of protein needs to get made, because maybe that mRNA sent out into the cytoplasm from the nucleus, but it's actually one that needs to be processed on a rough ER. So how do they get moved around? Okay. Next, I'm going to cover a little more about protein folding. I think that's a good time to review that. This is something I just worked up, but you have this back in your notes. This is all review from the beginning of the year when we did biochemistry and everyone was crying and struggling with polarity, but I thought I'd just go over it for you. You can look through your notes and former assignments if you like to. So we have 4 levels of protein folding: primary, secondary, tertiary, quaternary. Primary protein folding is just the sequence or the order of those amino acids. So it's what amino acids are connected to each other by peptide bonds. That was done here by showing different shapes and different colors, right? There's 20 different amino acid. What's the order? Secondary structure. I've got Dave opening a bag of cookies behind me. I wonder if you can hear that. Can I have a cookie, Dave?
Yeah.
Okay. All right. Secondary structure is not yet anything unique to each specific amino acid, but we are interacting between the hydrogens and oxygens and nitrogens of these repeating structures. So not anything to do with the R groups yet, but interaction with the repeating section of the protein backbone. That's where we get our alpha helixes and our beta pleated sheets, and this is happening again with just hydrogen bonding and forming repetitive shapes and structures of the protein. The unique stuff here comes in the tertiary structure. This is where now the unique interactions and foldings between the R groups happen. So this is now where all those 20 different amino acids had different kinds of properties, and now they're getting to interact with each other. Cysteines with sulfur are forming disulfide bonds, really strong connections that are holding big sections of the protein together. Acid and base reactions, positively and negatively charged amino acids are going to ionically bond together, again, holding the protein together. Hydrophobic ones are going to be on the inside. Hydrophilic ones are going to be facing on the outside. And then some general polar hydrogen bonding. If you've got some oxygens on something, some hydrogens on others, they might be interacting loosely with each other with hydrogen bonds. And some, but not all proteins will do the 4th level of protein structure, quaternary, where several protein units are going to come working together. Hemoglobin is a great example of this that has, I believe, alpha pieces and beta pieces, 2 and 2, not to be confused with alpha and beta pleated sheets, but like a beta hemoglobin and alpha hemoglobin. So that's a review of protein folding. And we want to recall this because if we have a mutation, we've got problems, right? So this is, again, something that I've just kind of worked up to review with you. It's all part of chapter 17. I would look at section 17.5 for an example if you want to see in the textbook. I don't know exactly what page that is, but 17.5 is the section. So here is my original DNA message. I've got my DNA in blue, my RNA in teal, and then my resulting amino acid protein structure here in like fuchsia. So you can also find an amino acid expression table on page 330 in the 9th edition, or you could just Google codon table. There's also going to be one on the assignment that I'm posting, but if you want to just work off online, you can do that. Look up codon table. You can find the wheel version if you like that, or the rectangular one. On the AP Bio test, it will be the rectangular table-like structure, not the wheel. So we've got our original template strand DNA, and then I'm going to be transcribing the RNA off of that. So remember and recall from transcription that RNA is going to be built 5' to 3', and it is working off of the template strand in the reverse order, 3' to 5'. So if I'm going to build this RNA 5' working to 3', that means I'm reading the template strand in reverse. Okay, so I don't know if the AP will get this complicated. Definitely if you're in genetics, Ms. Pagels has been doing this, but I'm just going to include it here so that we're doing everything correctly. So as I read along the DNA, I'm not looking for the start codon. If I am transcribing, I'm already starting back here where the promoter would have been, and I'm working my way up to where the gene gets encoded. Like this would be the start of the gene, okay? But there is always a little bit more upstream of that. So don't just assume you're going to start coding your amino acids right at the beginning with this GGC codon. Look in your RNA where you find AUG. That's where you start translation, okay? But your transcription would have occurred upstream and downstream from that. So remember, every time you're needing a T to be complementary to an A, you're going to use a U. And so I've worked my way across, and I've highlighted here what is my start and stop codons just so you can see them, because I put them there and I know they're there. So AUG is our start codon, and I'm actually going to pull up a chart for you if you don't remember how to code. Stand by. Oh, nope. What? Oh my goodness. Technical difficulties on my iPad. Codon chart. Okay. So, if I have, where am I? Up here. AUG, we already know that that's methionine, but let's say CAU. How did I find out that that's histine? Well, here on your chart, you're going to do your first letter, C. So you find which row you're in. So we are going to be in this row. CA in this column, okay, second letter A. And CAU is the last one, so it's the first one. CAU is my codon. Histidine is my amino acid that I would use for that. So I worked my way across, and my original DNA message coded for a methionine, histidine, leucine, glycine, arginine, lysine, and then UGA is a stop codon. This doesn't count for an amino acid, so my original one, oh wow, I did it wrong. 1, 2, 3, 4, 5, 6 amino acids long to this protein. Okay. So now I've done some mutations. So in the textbook on 17.5, they mention a substitution or point mutation. This is the action of what is happening to the DNA. It means a change of one nucleotide base pair. So here I've changed AAT to AAC. And over here I did some others. I actually only wanted to do one of them. I ended up doing both because, you know, I messed up on my colors. So we're going to roll with it. Here I changed the third letter of the codon. So what was ATT coding for UUA now changes AAC and we get UUG. Well, UUG still codes for leucine because of the redundancy in the code. You can see UUA, UUG, you're getting the same amino acid. A lot of ones are proline as well. Arginine is a really common amino acid. And so you can still make a third letter substitution because there's redundancy in the codon table. You're not going to have necessarily a change in your amino acid. So what that would be called is a silent mutation. So what did we do to the DNA? We did a substitution or a point mutation, and the result could be a silent mutation. This is really common when you're changing the third letter of a codon, and it's due to that redundancy in the codon chart. So here I've also done substitution mutations where I've changed the middle letter and I got proline instead of arginine, and I changed the first letter and I got a glutamic acid, I believe, instead of lysine. So these are missense mutations. Missense mutations is when you've changed one letter in a substitution nucleotide and it results in a change of one amino acid. Yes, I happen to do 2 in a row to show you like you're changing maybe a first or a second letter. So now you ask yourself, how big is this mutation going to be a problem for the final protein? If you changed one to an amino acid that has the same kind of property, like is proline and arginine really similar in their chemical structure? You'd have to look up the molecular structure of those. Are they both nonpolar? Well, maybe it's not a big deal, right? But maybe you swapped a positively charged amino acid for a negatively charged one. That's going to be a bigger problem for the protein. Maybe you changed an essential part of the active site, and now the protein isn't going to be able to do its entire function. Recall that sickle cell anemia is a change of one letter in the DNA, and it changes like a glutamic acid to a valine or one to the other, and it changes and results in an entirely different phenotype for people. The other one here is if we had a change of a substitution, right, changing one of the DNA letters, but we end up coding for an early stop codon. So maybe we had a UGA over here instead, and we ended everything. That would be a nonsense mutation, okay? So we have, what are we doing? Substitution mutation. We can get silent, missense, or nonsense. These are kind of our outcome choices. So a nonsense mutation is when the codon for the amino acid is now a stop codon. Basically, your protein becomes non-functional because you haven't finished making it. If this happens really far towards the end of the protein, maybe it's not a big deal, but obviously if it's early, your protein is far limited in size and you don't have what you need. So our other form of mutations is insertions and deletions. So this is where you add or lose nucleotides, and it's a big problem because you get frame shifts, and that moves all of the codons over by 1 letter, 2 letter, here I've added 1, and you start having multiple missense mutations to the point where your protein is no longer what it used to be. You may even end up with a nonsense mutation. Here you can see I've gotten an early stop codon than the one that I intended to. So this is not only different, but shorter than my original protein was supposed to be. So that's mutations. No, I think that's about it. This is all in chapter 17.5 of the 9th edition of the textbook. I think it's probably still chapter 17 of the 7th edition, but I don't have one here at home to reference. So just look in the table of contents and you're looking for DNA and proteins, or DNA to proteins, or protein synthesis. So I wanted to explain to you our upcoming activity that I think we could do. By no means is this an assignment that you have to do, but you're probably bored, right? You probably want something to do and something to practice. So I have posted this assignment onto Schoology. It's called Speaking in Code. So what you're going to do is this is uploaded to the handout section on Schoology, and we're going to use what we know about going from DNA to RNA to proteins and using this coding of nucleotides to amino acids. We're actually going to go backwards. So your task is to create a message to others using the single letter abbreviations. So if you're looking at an amino acid table like this, it doesn't have the single letter abbreviations. I've been working with these long enough in my life that I know valine is substituted as a V if we're talking about single letters. Alanine is an A. Leucine, lysine, one of them's L, one of them's K. So I've actually posted this one on the assignment. It's got, leucine's L, lysine's K. Ha, I win. And so this has not only their full names, but it's got the single letter codes. And so you're going to start with the message you want to say. So what are you going to try to say to people? Are you desperate for a rescue, being quarantined and social distancing yourself? What are you doing to keep yourself busy? A new hobby you've learned, something like that. Let's keep it clean, and I will show you how to post onto Schoology in a minute, but I'm going to show you how to work backwards. So here's my message. It says, "Hi there." Can be short. It doesn't need to be too complicated, but it's just so you guys can practice basically reverse coding to really know that you know what to do going forward as well. So I know that H is going to be histidine, and one of the RNA codons for histidine is CAC. So I'm using that on the table. I find histidine, and I'm using any of these. I'm just going to use the second one. I is isoleucine. I'm going to use AUU. Okay? And I'm going to keep going here, but I'm going to let you watch what I'm doing. So threonine, ACU, histidine. Again, why not use the other one? CAU. Glutamic acid, GAA. Arginine, AGG, and another glutamic acid, GAG. So that's my RNA. And now, what would my DNA be to work backwards from here? Why not? Different colors, right? CAC, so G, T, G, A can bond with T, U will bond with A with T, C with G, A. Even I'm brain farting probably. I bet some of this will be wrong, and then you'll catch me on it. G with C, C, T, 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 C, 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 T, C. All right, I hope that's right. A's with T's, U's with A's. Great. So that's my message. This is what I'm going to upload to the discussion post. So while this video uploads and buffers on YouTube, I'm going to create this post. I'm going to put my new message in, so I'll create something else rather than just "hi there." But go ahead and post your DNA message, and then people, what I'd like you to do is once you've created your own code out there, don't give them this. That's what people are going to try to figure out. So they're going to take each other, go find someone else's DNA message, transcribe it into RNA, and then use the single letter amino acid expression table to figure out what's the hidden message. And we can talk to each other on Schoology through genetic code. Why not, right? If you want to get real technical, you can start with methionine, and that means your message has to start with an M, and in here would have to be AUG, but that kind of limits what you're able to say. And if you want to end with a stop codon, like UGA is my go-to stop codon, you can throw that in there too. But that would need to then be in the DNA. So a TAC and AC. TC, sure. But my hi there doesn't make any sense for me. So keep in mind, human alphabet only has 26 letters, but we have only 20 amino acids. So you don't have access to the letters B, J, O, U, X, or Z. So if your name starts with a J, sorry, you can't start with that. And we're missing 2 out of our 5 vowels. So that's going to be a little tricky. So there's the challenge in this activity. I won't set a due date on it. Just go ahead and work on that now and through the weekend. And I will also post some formative assessment in the AP classroom on central dogma and mutations. Next week, or through this weekend, we'll see. We're going to work on operons and more intense gene expression things. So this will be the next one. And I have an activity and a video for you to watch about some pretty cool fish that every year end up on the AP Bio exam. So look for this. On Schoology, it's going to be in handouts. So in the regular folder, handouts, and we're on Unit 7, DNA to Proteins and More, and it's speaking in code. So that has the directions and also the single letter codon table. If you love the codon chart that is the wheel, these are also available if you just Google search them. What I would personally avoid is the ones that go from DNA to straight to the amino acids. I really don't like how they've been using those, but somewhere out there you can usually find ones that have the single letter here. Like there's one that's got all the single letters as well as, oops, the 3-letter code, so it's got a bunch of options for you. All right. Oh, and it even has them coded for what's their property. That's a good review too. All right, well, I'll let you go, AP Bio. Hope you're doing great. My sweater is coming along, and otherwise I'm extremely bored, so I'll be looking to give you guys some more material to work on, and I hope you're looking forward to it as well. See you later. Bye.